the third research, maybe the most important one, was research, in vivo research of autoimmune encephalomyelitis. I know that this name maybe can feel you a bit awkward, but this is just the animal model of multiple sclerosis. Why multiple sclerosis? That's an interesting question. We could examine Karnozin Extra activity in many other diseases, but we decided to be multiple sclerosis. Well, I remember one day we were at the office working, and the phone rang, and one guy called us and told us, "Oh my God, people, you have no idea what product you have. I have primary progressive multiple sclerosis, and I was tied to bed. And after just few weeks of taking your supplements, I felt I have more strength. I could get up from the bed, and now I'm making my first steps." I have to confess that at that moment, we were kind of surprised by his story, and personally, I didn't believe it because I know that primary progressive multiple sclerosis is the type of multiple sclerosis that never gets better. It always gets worse, and there is no cure for primary progressive multiple sclerosis. But as we are living in a modern world where information is spread very quickly, it wasn't long before people started to call us and to telling us the same testimonies. We were receiving the same testimonies, and those were the people, let's say, from different parts of the country who didn't know each other. And at that time, we excluded coincidence, and we excluded so-called placebo effect. And at that time, we knew that we have something very, very important in our hands, but we didn't know what to do about that. Then we did some brainstorming, and we went to the medical faculty, University of Novi Sad, and turned to my professors. Together with them, we designed an experiment which included the mice. We induced multiple sclerosis in mice. And we applied for governmental research funding in order to get the money for experiment, which was very expensive. And actually, it was very encouraging when the government of Republic of Serbia realized the importance of this experiment, and we got the asked financial support from them. Before I show you result which we gathered out of this experiment, I would like to tell a few sentences about multiple sclerosis. Multiple sclerosis is a neurodegenerative autoimmune-mediated disease which affects more than 3 million people worldwide. On this slide, we can see the entire pathogenesis of multiple sclerosis. We will go through it. So here we have a neuron. Neuron had a body and a tail. The axon or the tail is covered with myelin sheet. Myelin sheet is made up of cholesterol, mostly of cholesterol, statins again, of cholesterol, and this myelin ensures that nerve signal is transmitted very quickly through the axon. This myelin is produced by oligodendrocytes. Oligodendrocytes are the supportive cells in the brain, and their only role is to produce the myelin necessary for quick transmission of information. What is interesting about oligodendrocytes? When I start to search for the reason why Karnozin Extra is helping the people with multiple sclerosis, I went through many scientific papers, and I remember I was reading one paper, and on the I don't know which page I found one sentence, and that sentence was very, very important to me. That sentence was, oligodendrocytes are the only supportive nerve cells in the brain that possess carnosin synthetase. They have an enzyme which is able to synthesize carnosin when it's not present out of 2 amino acids. And the cells are so smart. Cell wouldn't have something that it doesn't need. So oligodendrocytes, because they have carnosin synthetase, they need carnosin for some reason. And maybe that is to produce and synthesize the myelin. What is happening in multiple sclerosis? Immune cells, our immune cells, T and B cells, are attacking the myelin. And depending on where this myelin is destroyed, people who suffer from multiple sclerosis are experiencing different symptoms. Those symptoms can include vision disturbances, can include spasm, tremor, pain in the muscles, problem with coordination, problem with speech, sexual dysfunction, and so on. So basically, what is happening in multiple sclerosis? We have in the brain something that is called blood-brain barrier. This barrier led only certain substances to enter the brain. L-carnosin can easily penetrate the blood-brain barrier, but T and B cells cannot enter the brain. Once this blood-brain barrier is damaged, for example, because of oxidative stress or lipid peroxidation or whatever, T and B cells are entering the brain. And they are seeing the myelin, which they never seen before, and they are attacking the myelin. While they are attacking the myelin, all immune reactions are producing the inflammatory response as well. So while they are attacking the myelin, they produce a lot of inflammatory cytokines. So basically, multiple sclerosis is immune-mediated, but it's also inflammatory disease. What we tried to prove with our experiment, we wanted to prove that Karnozin Extra is able to reduce oxidative stress, it's able to reduce oxidative damage to blood-brain barrier, and consequently to suppress inflammatory reaction caused by immune response. That was the first thing we wanted to explore. And the second one was that Karnozin Extra is able to recover oligodendrocytes and help them to produce myelin again. The design of experiment was as following. So we had 3 groups of mice. In all 3 groups, we induced multiple sclerosis by using botulinum toxin. And the first group was just with multiple sclerosis. The second group of mice we were feeding them with dose that corresponds to 2 capsules in humans. And the third group was on 6 capsules, on the dose that corresponds to 6 capsules in humans. We fed them for 25 days. In mice lifespan, that is 25 days, and that corresponds to 2.5 years in the human lifespan, which is sort of long-term administration of Karnozin Extra. What we have observed after 25 days. The control group without carnosin and the second group that was on the dosage of 2 capsules per day. Those 2 groups were identically sick. They couldn't move. But the third group was moving. They were not running a marathon, but they were moving. So what we have observed? Then we sacrificed those mice and look into their brains. We look under the microscope, and what we have observed? First, there was no observed difference between first and the second group. The third group has less demyelinating areas and less inflammation infiltrate per day, comparing to the control group. So we had less demyelination and less inflammatory response in those brains, meaning that dosage of 6 capsules per day were enough to slow down the process of developing the multiple sclerosis. And when we sacrificed those mice, we wanted to see if there is some. We knew that Karnozin Extra is not showing any side effects, but we wanted to prove that on a molecular level. So actually, we took the samples from all of the tissues, from liver, from gut, from kidney, brain, heart, spleen, pancreas, and testes. Look under the microscope to see if something is happening after the long-term administration of high dosage of Karnozin Extra. We didn't find any histological, morphological, or functional changes in those tissues when high doses of Karnozin Extra were used. And these findings suggest that we have highly safe product.